Hi, everybody. So, welcome. This is our last session for this year, and it's my absolute pleasure to introduce a senior graduate student in the lab named Jessica Weiselberg. I will say I wasn't even looking at the schedule, and to Jessica's tremendous credit, a couple of months ago, she called me and said, "There's one slot left. Do you think it would be okay if I gave it?" And I said, "That is fantastic. Go for it." So, I'm very proud of her for taking that initiative and feeling great about her work, such that she'd like to share it and get input. I will note that Jessica joined my lab, and exactly one week later, the world closed. And then Einstein stayed closed for 4 months because of the COVID pandemic. And so we met remotely for 4 months, and she really made a lot of stride in reading and thinking and learning about techniques by paper. And then she ultimately got to come into the lab and do that. And she's really produced a beautiful body of novel work about roles of the effects of methamphetamine on human HIV-infected macrophages. Jessica's going to be defending her thesis in August. And so any input or questions you could give would be really helpful and fun at this time. And she really enjoys it. She's a terrific addition to the lab. We will miss her, but I'm really excited about her work and her next steps. So I don't want to take up all your time, so go ahead.
All right, let me just get my screen set up. 1 second. Mhm. Okay. All right, good afternoon, everyone. I'm so excited and so honored to be here with you this afternoon and to tell you about my thesis work. So, this audience knows very well that HIV remains a global health problem, with approximately 40 million people worldwide living with HIV as of last year, and with over 1 million new infections. So the epidemic is ongoing. HIV is treated with antiretroviral therapy, or ART, which successfully suppresses viral replication and prevents the progression of infection, but it does not eliminate the virus and thus is not a cure, as the virus establishes tissue reservoirs which are difficult to eliminate and target with ART. One such reservoir is in the CNS, and HIV enters the CNS really early after peripheral infection, often long before an individual even knows that they've become exposed to the virus. And HIV entry into the CNS results in the manifestation of HIV-associated neurocognitive impairment, or HIV-NCI, which affects up to 50% of people with HIV despite adherence to ART. And it manifests as a heterogeneous spectrum of cognitive and motor deficits that significantly impair quality of life. So, our lab and others have implicated a subset of monocytes as participating in HIV-NCI pathogenesis, as these monocytes are elevated in the peripheral circulation of people with HIV and have also been shown to be preferentially infected by the virus. These monocytes are characterized by high expression of CD14, the LPS co-receptor, and CD16, the Fc gamma 3 receptor. These monocytes are sensitive to chemokines, including CCL2, which are elevated in the CNS of people with HIV and are presented on the blood-brain barrier endothelial cells. Monocytes cross the blood-brain barrier in response to these chemokines, where they differentiate into macrophages, establishing the CNS tissue reservoir and serving as long-lived viral reservoirs. These macrophages also shed virus and infect other parenchymal cells, including microglia and astrocytes. And in addition to infection, there's also activation of other parenchymal cells. So not every single cell is going to be infected with the virus, but there's also exposure to the virus and viral proteins. And there is production of cytokines and chemokines and other host and viral neurotoxic factors, including reactive oxygen species. And in combination, all of these factors accumulate to a chronic low-level inflammatory environment in the CNS that ultimately damages neurons and accumulates and manifests as HIV-NCI. Concomitant with the HIV epidemic, substance use is a growing public health problem. And for this work, I'll be focusing on methamphetamine use. And Meth has been increasing over time in its use, with over 2 million people reporting use in the US in 2023 alone. Meth is a potent CNS stimulant, and chronic use can lead to neurotoxicity that results in cognitive impairment. The overlap with HIV is that meth use has been associated with an increased risk of HIV acquisition, as well as detectable viral loads in people with HIV who use meth, even in the presence of stable art. There appears to be an increased prevalence of meth use in people with HIV, and a recent cohort study in the US showed that 17% of the cohort of people with HIV were people who use meth. It's important to note that studies have shown that some people with HIV who use meth chronically have increased neuronal damage and neurocognitive deficits compared to people with HIV who do not use meth. So the questions that the lab is interested in is, how does methamphetamine impact monocyte migration into the CNS? As for the purposes of my work in this talk, I'll be focusing on the macrophages once in the CNS established as the tissue reservoir. How does methamphetamine impact their function? So why macrophages? Macrophages are important immune cells, and they also, once again, serve as long-lived viral reservoirs in the central nervous system. Macrophages perform many different functions, all aimed at maintaining tissue homeostasis, and wherever they are in this context, of course, once again, we're talking about the brain. Macrophages produce many enzymes, proteases, and Mendel-binding proteins, which are both intracellular and secreted. Macrophages also produce reactive oxygen species, which actually play a role in normal cell signaling, but when aberrant, leads to oxidative stress and neuronal damage. Macrophages are also antigen-presenting cells, and they interact with the adaptive immune system and facilitate those responses. And macrophages also, of course, produce cytokines and chemokines, which are also an important part of normal CNS homeostasis and normal levels of immune surveillance in the CNS. But you can imagine that when any one of these functions or any other macrophage functions not shown here are dysregulated, whether increased when they shouldn't be or decreased when they shouldn't be, these cells could be contributing to HIV and CI by not being able to properly perform those homeostatic functions, and therefore could lead to neurodegeneration and HIV and CI. The lab has previously published some work on the impact of methamphetamine on macrophages, and some findings highlighted here amongst others. We showed that meth increases reactive oxygen species production by HIV-infected cells, and in a proteomic study of HIV-infected macrophages treated with meth, the top pathways by IPA that were impacted by meth were those involved in Parkinson's, Alzheimer's, Huntington's, and prion disease processes, really highlighting for us that meth is imparting a pro-inflammatory, pro-neuroinflammatory, and neurodegenerative-like phenotype in these macrophages treated with meth, which highlights a further connection to HIV and NCI and worsening NCI in people with HIV who use meth. And you can see that the title of this slide identifies that this was done with a 24-hour treatment. And for my studies, I wanted to model more long-term use, or longer-term chronic, however you want to describe it, use, given that people with HIV typically do so on a more regular basis, rather than just one 24-hour treatment. So we hypothesize that longer-term exposure to meth results in alterations in macrophage functions that can contribute to HIV and CI. And for the purposes of this, we establish a longer-term exposure with a 5-day daily treatment. And I'll show you that in the coming slides in the rest of the talk. So to address our hypothesis, we will characterize the impact of longer-term meth treatment on the transcriptome of uninfected and HIV-infected macrophages. I want to highlight that we did do sequencing on uninfected macrophages, but I won't be focusing on that in this talk. And then, of course, evaluate the contribution of those transcriptional changes to functions in those macrophages that could contribute to HIV and CI. The way that we obtain our macrophages is by obtaining leukopaks of healthy individuals and using Ficol overlay and density gradient centrifugation. We isolate PBMC. And then the way that I obtain my macrophages is kind of one of two main culture strategies that I'll go through. And depending on the downstream assay type um, determines which one of these methods I'll be using. So I either isolate PBMC and then plate those PBMC in total, or I do negative or positive monocyte isolation from the PBMC and then plate those isolated monocytes on 24 or 96 well plates. And then with those isolated cells, we culture and mature with macrophage colony stimulating factor, or MCSF, for 6 days. And that allows the monocytes, whether from the whole PBMC or the isolated monocytes, to adhere to the plate and differentiate into macrophages. And then at the end of 6 days of culture, we have our mature differentiated macrophage population that we go ahead for downstream experiments with. So the first thing that we wanted to establish was that we could model a longer term meth treatment in vitro, as we had not done this prior. So to establish that, we took macrophages that were isolated, as I just described, differentiated for 6 days, and then infected with HIV. And then we allow 2 days to propagate the infection. And after 2 days, we begin daily meth treatment for 5 days, such that at the end, we have HIV infected untreated cells and HIV infected meth treated cells that were treated for 5 days daily. And then I collected supernatants, which was used to measure HIV P24 by alpha ELISA to measure infection levels, and LDH-XA to measure cell toxicity. So this is data from P24 alpha ELISA showing that meth does not change HIV infection levels. The graph on the right is from supernatants from 17 independent donors, and we do not see a difference in infection levels at the end of 5 days of treatment, nor at each day of the 5-day daily time course across the different time points. So the fact that we don't see any changes in P24 levels indicates that any changes we do find are not because meth is altering infection levels in our macrophage culture system. And next, looking at cell toxicity, we show that meth treatment is not toxic to cells at any of the 5 time points. This is LDH assay data or cytotoxicity assay data with LDH release represented as percent of max on the y-axis. And the way that this is done is by we collect supernatants and then lyse the cells underneath, as LDH is something that is an intracellular enzyme, and if is extracellular in your supernatant, it means that there's cell death. But the cells are used and are lysed and used as a positive control, as lyse cells will release LDH. So we use that as 100% lysis, and then the supernatants are normalized to that 100% lysis. And what you can see is that we do not see an increase in cell death with meth treatment at any time point. There are some donors that do have more cell death than others, but what you can notice is that there's no difference with meth treatment, and therefore meth is not toxic to any two cells at any ever 5-day time point. And with these data, we show that meth does not change infection levels, nor is toxic to cells, and we conclude that any changes that we do find indicate that meth is altering macrophage cell biology and not changes to infection levels or cell death mechanisms that might otherwise be upregulating other mechanisms. So it's purely the impact of meth on macrophage cell biology rather than these two other factors. So now to characterize changes to macrophage transcriptome, we performed RNA sequencing with macrophages from 5 independent donors, differentiated for 6 days once again, infected with HIV, and then treated with meth daily for 5 days, such that at the end we have untreated cells and meth-treated cells for 5 days, collected supernatants once again for P24 alpha ELISA, and extracted RNA, which was sent to Novogene for sequencing. And we found many different changes in many genes that we grouped by macrophage functions. And the sequencing was done by Drs. Meng Nu and I'm sorry, not the sequencing, the data analysis was done by Drs. Meng Nu and Howard Fox at the University of Nebraska Medical Center. So a huge thank you to them for the analysis. Um, and among the many different macrophage functions that we identified gene changes in, we chose to pursue a family of genes that we identified upregulation in, known as metallothionein 1. And I'll explain more about it, of course, in a minute. But the reason that we were interested in these genes is that, number one, they have roles in neuroprotection as well. The protein encoded by these genes has roles in neuroprotection, but also neuroinflammatory processes. So we thought that duality was very interesting of the protein. And also, this protein has not been characterized in HIV-infected macrophages, nor in the context of substance use. And this is entirely novel, and we are interested in pursuing these studies. So this table here is a representation of those gene expression changes for 5 independent donors that we sequenced. The gene names on the left are all gene isoforms of MT1, metallothionein 1, and the right is a log2 fold change values for meth-treated cells compared to untreated cells. So we see increased expression of the MT1 isoforms X, H, F, G, and E. So a little bit about metallothionein. Metallothioneins, or MTs, play an important role in protection against oxidative stress, heavy metal toxicity, DNA damage, and apoptosis. There are 4 MT proteins labeled MT1 through 4. MT1 has many gene isoforms, as I showed you on the previous slide, which we have been working with. We also did see changes in increased expression of MT2 gene, but we chose not to pursue that for right now. So a little bit about its function. MT1 in the cytoplasm neutralizes ROS. And during oxidative stress, actually, MT1 will localize to the nucleus and result in upregulation of its own transcription, upregulation of other ROS response mechanisms, and cytokine production in some cell types due to interactions with NF-kappa B. So there are some established connections between MT1 and inflammation and neurodegeneration, as it's been shown to be increase in other neurodegenerative conditions, as well as in acute brain injury. And this literature implicates MT1 as neuroprotective to deal with CNS damage. MT1 expression has been shown to be upregulated in microglia treated with amyloid beta, and it was shown that MT1 expression has been associated with A beta plaques in Alzheimer's mouse models. And it's important to know that A beta abnormalities are part or found in people with HIV. And people with HIV and CI, particularly. There's a little bit known about MT1 and monocytes and macrophages. One big thing about MT1 is that it binds metals, including zinc. And zinc is important for immune cell signaling and functioning. And so MT1 in different immune cells is a big topic of interest in the literature. There was one study that showed that MT1 was upregulated in HIV-infected monocytes, and they linked intracellular zinc levels, again, MT1 binds zinc, to apoptosis resistance in HIV-infected monocytes, and that, of course, apoptosis resistance is a big phenotype of infected monocytes. Once again, relating to the nuclear localization, MT1 regulates NF-kappa B activity in some cell types it activates it, and in some cell types it actually results in negative regulation of NF-kappa B activity. So, and when it is activated, it results in cytokine expression in certain cell types. And it was shown in MT knockout mice, macrophages treated with LPS from those mice produced less TNF-alpha and IL-6, linking MT to cytokine production in macrophages particularly. And I want you to keep that finding in the back of your mind because I'll be talking about it later on in the functional studies. So the next thing that we did was validate our sequencing findings by QRT-PCR. And to do that, we have macrophages, once again, isolated and differentiated for 6 days. And then after 2 days, we began daily meth treatment for 5 days. At the end of those 5 days, We collected supernatants again for P24, alpha lysa, and then we extracted RNA, converted into cDNA, and did qRT-PCR for our MT1 genes of interest. So here are the qRT-PCR data for 9 independent donors for our MT1 genes of interest, X, H, G, F, and E. And using the 2 to the delta delta CT method, and we use 18S RNA subunit as our housekeeping gene. We represent the data as relative mRNA expression level with untreated cells set to 1 and meth as a fold change value compared to that. And you can see we have significant increase in MT1X and a trend towards an increase for H, G, F, and E for 9 independent donors. And given that these are primary cells and there is variability among donors, we were curious to see how across the 5-day time course the different donors respond. Rather, at one snapshot in time, we wanted to see how the gene expression levels changed across the 5 days of treatment. So we did a time course PCR kinetic assay. And to do these experiments, once again, with macrophages isolated, differentiated, infected with HIV, and treated with meth. But this time, after each day of treatment, we collected supernatants, extracted RNA, cDNA, qPCR. So after day 1, 2, 3, 4, and 5, we did this to analyze the kinetics of MT1 gene expression changes with meth treatment. And to no one's surprise, these are primary cells. We did not find a particular time point of greatest increase in the genes. There wasn't like a consensus time point in all the donors. So we choose to represent the data as maximal expression level. So the way that we did this was for each donor, for each gene, we identified the greatest gene expression increase over untreated for each donor, and then use that value as represented in each of these plots. So, because there wasn't a consensus time point, we chose the value of greatest gene expression increase for each donor for each gene, and that's what's represented here. So we have relative mRNA expression level with untreated set to 1 on the y-axis once again. And for 10 independent donors, we see significant increases in MT1X, H, G, F, and E. So now that we've validated our RNA sequencing findings and identified how the donor expression levels change across the time points, we wanted to understand, do those gene expression changes correlate with functional changes with meth treatment as well? So the first function we chose to evaluate was nuclear localization of MT1. As I mentioned at the beginning, that it's important for upregulation of other ROS response mechanisms, but it also can result in cytokine production. So we wanted to understand how meth treatment changes nuclear localization. And to do that, we positively isolated monocytes and then plated the cells on glass cover slips and differentiated for 6 days with MCSF once again. We infected with HIV and then treated with meth daily. And one thing that we came to appreciate from the time course gene expression assays is for the functional studies, we chose to do them all in a time course as well, seeing how differently the donors respond across the time points. We chose to do all the functional assays in a kinetic as well. So after each day of meth treatment, we fixed our cover slips and stained for MT1. And then we imaged the cells by confocal microscopy, and this allowed us to evaluate the MT1 that is within the nucleus. So here is representative images from a donor, untreated, and its paired meth treatment. And we show that meth treatment increases nuclear localization of MT1. So you can see here the nuclei is stained with DAPI, and then MT1 in green. You can see that there is a lot of MT1 in the nucleus, as indicated by these puncta here. And there's a little bit in this one untreated nucleus. It's not negligible. It is there. This nucleus really doesn't have very much, but you can see that there is more of it. And then we stained for actin to outline the cell cytoskeleton. using phalloidin. And then on the merge, you can see that we have more nuclear localization of MT1 in the meth-treated cells compared to untreated. And the theme of a lot of this work is, I've said a bunch of times already, donor variation across the time course. Um, so I'm going to show you in the next slide how we see changes at different time points across the time course in all of the donors. So here are the plots that I used to the quantification of the images that I just showed you. So I'll focus here on just one, but we did this with 9 independent donors. And on the y-axis, we have nuclear signal intensity. So, mean fluorescence intensity per nucleus. Each dot is a singular nucleus. The conditions in red are all the untreated conditions, and the blue is the meth-treated conditions for all 5 days across the time course. And you can see that we have increases, a large increase here, and sometimes no change. And you can see sort of, if you look across the different plots, how things are fluctuating in the different donors and how things are changing. So we performed a similar maximal expression analysis or maximal localization, maximal increase in localization, as we did with the gene expression studies. And to do that, we generated a full change value. So you see the lines in each condition represent the mean fluorescence intensity. We generated a full change, so meth divided by untreated, where untreated is set to 1, so a full change of meth treatment. And we chose for each donor, the maximal increase in localization, because you do see time points where there's multiple time points of localization, but this is obviously greater than this one. So this is what was chosen to represent that donor. And I'll show you in the following slide the pool of all these values. So here we show the full change values with untreated set to 1 in nuclear localization of MT1, again representing the mean fluorescence intensity represented as a fold change. And for 9 independent donors, we see increases in MT1 nuclear localization with meth treatment. So given that we've seen this increase in nuclear localization, does that localization correlate to cytokine production? Which was our next question. And to do these experiments, we positively isolated monocytes, plated and differentiated for 6 days with MCSF, infected with HIV, and then treated with meth daily once again. And for these experiments, we wanted to understand if meth, in the presence of another inflammatory stimulus, how that would change cytokine production and if it would further increase it, or if meth alone doesn't stimulate cytokines, but meth plus the second stimulus would stimulate cytokine production. So we chose LPS for these studies, and that was done for a few reasons. Number one, LPS was used in the mouse empty knockout macrophage study that I talked about earlier at the top that I asked you to keep in the back of your mind. So it was shown that LPS treated macrophages from empty knockout mice produce less cytokines. So that was a tie in there. Also, LPS is a biologically relevant stimulus as LPS is elevated in the blood and CSF of people with HIV. So first, we actually optimized the treatment kinetics for LPS. And this was done by a really talented graduate student, rotation student that I had last year, Tanya Hymas. So we identified that 1 nanogram per mil LPS for 3 hours was optimal in our assay. And so the way that we set up our treatments is that we did meth treatment for 24 hours and LPS treatment for those conditions for the last 3 hours of those 24 hours. So at 21 hours, the LPS was added. And then at the end of the 24-hour time point, we collected all the supernatants. So our final conditions were HIV untreated, LPS treated, meth treated, and then meth plus LPS. And then we collected supernatants, aliquoted, and assayed cytokine production by Luminex multiplex assay. And this was done by the CFAR biomarkers and advanced technologies core here at Einstein. So the inflammatory mediators that we assayed were identified from a recent meta-analysis that looked at different markers in the CSF of people with HIV and correlated and identified markers that correlated either with HIV NCI or general CNS markers of inflammation elevated in people with HIV. So the panel on the left, this is all one panel, but we broke it down by these two measures. So the markers that were correlated with HIV NCI that we analyzed were TNF alpha, IL-6, CCL2, CCL8, CXCL10 or IP10, CX3CL1 or fractalkine, and CXCL8 or IL-8, and SPP1 osteopontin. And the markers of inflammation elevated in people with HIV that we chose were CCL7, CCL5 or antes, CXCL9 or MIG, and CXCL12 or SDF1. So just before I get into the data, our samples had a lot of CCL2, and at all the different dilutions we tested were all above the limit of quantification for our assay. So we don't have reliable data for CCL2 because our macrophages just make a lot of CCL2, as you can imagine. But also similarly for IL-8, we saw the untreated and meth-treated cells also produced a lot of IL-8 that in a lot of the conditions was above the limit of detection as well. So I'll only be showing data for meth plus LPS because those were done at different dilutions and we're able to be better captured on the curve. And we also, fractalkine, CCL5 rantes, and CXCL9 MIG were below the limit of detection. So we don't have data for those either. So here are the data. So we see that meth treatment with and without LPS appears to increase inflammatory mediators shown to correlate with HIV NCI. So first we have here IL-6, TNF alpha, and IL-8. And we did the same maximal production analysis method as we've done prior. So across the 5-day time course, we chose the value of greatest fold change in production for each mediator with meth compared to untreated and with meth plus LPS compared to LPS alone. It's important to note that all of the donors at all the time points responded to LPS, and we're just showing the data as meth plus LPS compared to LPS alone, but know that they did respond to LPS, and this is on top of LPS treatment levels. So now actually getting into the data, we have on the left here IL-6 meth compared to untreated, and we see an increase in IL-6 production, and then a further increase in IL-6 with meth plus LPS compared to LPS alone. And same for TNF alpha. We see an increase in TNF treatment with meth compared to untreated, and then with meth plus LPS compared to LPS alone. It's important to note, you can see this pink and green donors just made a lot of TNF and IL-6, both in response to meth alone and then further on top of meth plus LPS. So you can see there is a lot of variability in the cytokine production, but we did see a really robust response for those two donors in a few of the cytokines. And for IL-8, I mentioned in the previous slide, we saw for the meth-treated and the untreated conditions, the cells made a lot of IL-8. So two of the donors were above the limit of quantification, and we're not showing the data for just those two, the other two donors that we do have data for. But we do see a significant increase in IL-8 production with meth plus LPS compared to LPS alone. And then for the other mediators, we see a same sort of trend. So for CXCL10, we see an increase in production with meth treatment compared to untreated, and then a further production in CXCL10 or IP10 with meth plus LPS compared to LPS alone. Same for CCL8. We see an increase in CCL8 with meth treatment, and then a further increase with meth plus LPS compared to LPS, which is significant. And once again, for osteopontin, we see an increase in osteopontin with meth treatment compared to untreated, and then once again with meth plus LPS compared to LPS alone. So we show here that meth with and without LPS increases inflammatory mediators shown to correlate with HIV NCI. And then for the last two mediators, which are general CNS inflammation mediators found in people with HIV, we see the same trend again. We have CCL7 increase with meth treatment compared to untreated, and then a further increase in CCL7 with meth plus LPS compared to LPS alone. And then same for CXCL12, we see an increase, and then a further increase in CXCL12 with meth plus LPS compared to LPS alone. I want to highlight, you can see there's only an N of 3 for CXCL12, and that is because one of the donors for all the treatment conditions was below the limit of detection. So we only have data for 3 donors here. So we showed that meth with and without LPS increases inflammatory mediators that are both correlated with HIV NCI and are also CNS inflammation mediators elevated in people with HIV. And this suggests that meth could be exacerbating HIV NCI by that mechanism. And further, especially because LPS is found in the CSF of people with HIV, macrophages would be exposed to that LPS. Macrophages in the CNS exposed to methamphetamine in a person with HIV who uses methamphetamine could further be exacerbating HIV NCI, potentially by increased expression of and production of these markers. And the last piece for the MT story, as MT's one of MT's major functions is ROS production. We measured ROS to see that correlation. So we positively isolated monocytes, plated them and differentiated for 6 days with MCSF, infected with HIV, and then treated with meth daily. Once again, after each day of treatment, we measured ROS levels. And to do this, we use the DCF assay, which DCF is a cell permeable dye that when diffuses into cells and in the presence of reactive oxygen species becomes fluorescent and then is not able to diffuse back out of the cell. So the signal is lasting for a few hours. And then after incubation with the dye, we treat with piocyanin and hydrogen peroxide as positive controls. And then we quantify fluorescence in a plate reader. So we do not see overall changes in ROS levels for our 4 to 6 donors that we assayed. As you can see by the mean values here at the bottom, but the data are shown as full change over untreated cells set to baseline of 1. So overall, there are no changes, but if you look, we see a couple of donors here at day 1 and one donor at day 2 that actually have decreased levels in ROS below baseline. And we hypothesize there that MT1 and other ROS response mechanisms are effective at these time points and able to neutralize ROS to those levels significantly below baseline, substantially below baseline. And then with later time points, such as at days 4 and 5, and this one donor at day 3, you can see that there's increased ROS levels here. And we hypothesize that with additional exposures to meth, the ROS response mechanisms are no longer effective at neutralizing ROS. And we see an increase in ROS levels with the additional hits of meth treatment that, again, are no longer able to be neutralized with, and the neutralization mechanisms are sort of, spent, as it were, at this time point. In summary, we demonstrated increased expression of MT1 gene isoforms, MT1X, H, G, F, and E in HIV-infected macrophages treated with meth. There was increased nuclear localization of MT1 with meth treatment. Meth with and without LPS increased production of IL-6, TNF-alpha, IL-8, IP-10, CCL8, osteopontin, CCL7, and CXCL12. And we saw decreased ROS levels with meth treatment at early time points and increased ROS levels at later time points. So shifting gears a little bit from the MT story, I'm going to tell you now a little bit about some other studies that I've been working on, following up from the RNA sequencing that we did. And we identified by RNA sequencing gene expression of several surface markers that were changed in HIV-infected macrophages treated with meth. So this is the same table that I showed you up top with the MT genes. So we have surface marker genes on the left hand of the graph and log2 fold change values for meth-treated cells compared to untreated for our 5 donors. And the markers indicated by the blue boxes, the ones that I'll be talking about today. So we saw increases in VCAM1, ICAM3, CD52, and CD28 that I'll be talking about. And VCAM1, ICAM3, and CD52 were all upregulated, and CD28 is downregulated in the sequencing data set. So ICAM3 and VCAM1 are surface markers not well characterized in macrophages, most often thought of as being expressed by endothelial cells. And both ICAM3 and VCAM1 are expressed on brain microvascular endothelial cells, which make up the blood-brain barrier, of course. And they play a role in facilitating monocyte transmigration by facilitating the firm adhesion step of transmigration. So we have our astrocytes and our endothelial cells. We have CCL2 presented on the surface that binds to CCR2 on the monocyte, and that results in inside-out signaling that opens up the integrins LFA1 and VLA4 on the monocytes that can then bind their cognate ligands, ICAM1 or ICAM3, and VCAM1 respectively on the endothelial cells, resulting in firm arrest of the monocytes. And it was shown in one study that VCAM1 was increased on cerebrospinal perivascular macrophages in an EAE mouse model of MS, which suggests that they can play a role in facilitating extravasation of immune cells, which is diagrammed here. Once again, we have our astrocytes, our endothelium, our monocyte firmly adhered to the endothelium through interactions with VLA4 and VCAM1 and LFA1 and ICAM1 or 3. As the firm arrest step, but later down the line, we have the diapedesis step, where the monocyte is squeezing its uropod through the interendothelial junctions and through facilitation of junctional adhesion molecules interacting on the endothelial cell and the monocyte, opening up in a zipper-like mechanism, the monocyte will squeeze its uropod through the interendothelial junction. And then you have that perivascular macrophage with its ICAM3 and VCAM1 that can bind LFA1 and VLA4 respectively on the monocyte and sort of shepherd the monocyte through the endothelium and, as it were, lend a helping hand and bring other immune cells into the CNS. And this is really important if we're talking about HIV and CI. If these macrophages treated with meth have increased expression of these markers, they could be facilitating further immune cell recruitment into the CNS through this mechanism, which is why we were interested in these genes. So we have PCR data, same as the beginning of the talk with the MT1 story. So at the end of 5 days of treatment, represented on the left of the slide, so ICAM3 and VCAM1 assayed at the end of 5 days of treatment with relative mRNA expression level with untreated set to 1 on the y-axis. And we see a non-significant increase in ICAM3 and VCAM1 with meth treatment. And then using our same maximal expression analysis that I talked about earlier with, once again, relative mRNA expression level with untreated set to 1, we see significant increases in ICAM3 and VCAM1. And then two more markers I'll be talking about are CD28 and CD52, which can dampen immune cell responses. CD28 is kind of, I think, classically thought of as a T cell marker that normally results in activation, or signal 2 as it's referred to in the T cell world. So you have the MHC class 1 bound peptide presented by the antigen presenting cell bound to the T cell receptor, and then you have CD28 on T cells, which binds to CD80 or 86 on the antigen presenting cell, resulting in activation of the T cell. But it was actually shown that CD28, when expressed on macrophages, can bind to CD28 in T cells and negatively regulate activation, as shown here. So it sort of competitively inhibits for CD28 and doesn't allow that activation signal and actually results in downregulated activation of T cells. And by a similar mechanism, CD52 on antigen-presenting cells can bind Siglec-10 on T cells, and that negatively regulates activation as well. So we were interested in both of these markers, given that if meth is increasing expression of these markers, it could suggest that meth is downregulating the adaptive immune response. So Once again, qPCR data. On the left, we have the end of 5-day treatment for CD28 and CD52, and we see a non-significant increase in CD28 and CD52 expression for 10 independent donors. And then using our maximal expression analysis, we do see significant increases in CD28 and CD52. I want to point out that for CD28, we actually noticed that there was a significant decrease in expression as well. So the same way that there are points of significant increases when you pool the values that are less than 1, we also see a significant decrease in CD28 expression. And that actually is in line with a study that showed that in people with HIV who use meth, there is increased T cell activation and increased T cell exhaustion, suggesting that there's this heightened T cell response or exhausted phenotype. So the decreased CD28 actually is more in line with that literature, implying that meth is downregulating its expression, therefore leading to less negative regulation of T cells and therefore more activation. So we're not really sure what to do with these two conflicting pieces of data. And we'd love some input and thought into it because we're just not really sure what to do with both the decreases and increases because both are biologically relevant conclusions to draw. So in summary, our preliminary data for the surface markers indicate that meth increases expression of ICAM3 and VCAM1, enabling them to facilitate recruitment of immune cells into the CNS. We see that meth increases expression of CD28 and CD52, which negatively regulate T cell activation, potentially resulting in decreased adaptive immune responses. But at the same time, meth also decreases CD28 expression, which may result in increased T cell activation found in some people with HIV who use meth. So some future studies for overall, we want to do brain tissue staining for MT1 in meth-treated animal models, chip sequencing to identify, or chromatin immunoprecipitation sequencing, ChIP-seq, to identify which genes MT1 and NF-kappa B is interacting with. For ICAM3 and VCAM1, we want to do immunofluorescence staining, and then tissue staining to identify localization of those macrophages. And if the perivascular macrophages have increases in these markers, they could be facilitating the intravasation of cells, as I talked about earlier. We want to do immunofluorescence staining for CD28 and CD52, and then a functional T cell activation assay with autologous T cells from our leukopaks to measure markers of activation, including cytokine production, to see how meth is changing T cell activation. And then some overall future studies are characterizing gene expression, other genes identified from the sequencing, and their downstream functions. And then, of course, we didn't do these experiments with antiretroviral drugs, so we want to see how that would change with antiretroviral drugs. Some limitation of our studies is that we cannot get enough cells from one leukopak to do all experiments at all the time points, and therefore we can't make direct correlations among all of our functions. We did not optimize our treatment kinetics for the LPS treatments for each cytokine, so we didn't necessarily optimize that. For our immunofluorescence, we weren't able to distinguish between HIV harboring and HIV exposed cells, and that's a really important distinction to make to see if MT1 changes in those two populations. And we, of course, did not perform our experiments with ART. So overall, the transcriptional and functional changes we found indicate that meth may be contributing to HIV and CI pathogenesis, and we identified new mechanisms that can be examined further to characterize macrophage dysfunction in people with HIV who use meth. I want to thank Joan, who's been a wonderful mentor for the last 6 years, the entire Vermin Lab. A huge shout out to Tina Calderon, who's been a big part of this project as well. Past lab members, rotation students, including Tanya Hymas, who did the LPS optimization experiments, Howard Fox and Mung Nu for doing the RNA sequencing analysis, the C4 biomarkers and advanced technologies core at Einstein for the Luminex, the people at Novogene for helping us with our sequencing, analytical imaging facility at Einstein who helped me with all of my microscopy, my student advisory committee, the Department of Pathology at Einstein, which we're a part of, the Einstein Graduate Division, and our funding sources. And before I take any questions, this is a giant macrophage synthesis that I found. on one of my slides, and I just wanted to show it because it's really, really pretty, and just to show you how big some of these cells can get. I unfortunately didn't quantify this because it's kind of hard to quantify each nuclei when some of these nuclei are sitting on top of each other. But anyway, happy to take any questions. Thank you so much for listening.
So Leah, do you want to direct the questions?
Oh, I thought you were going to moderate, Joan.
Oh, okay. I introduced. I'm happy to moderate.
Yes, you're the
Uh,
moderator.
okay, Jessica, there are some questions in the chat. So Marcus, do you want to ask your question? Hi.
Hi. Okay, thank you. Jessica, wonderful work. Really, really nice and very nice presentation. It's just so there's a lot a lot of questions, right? That comes. I just some of the basic questions. So did you look at, sex dependent differences? Did you see any, or do you know what your donors, how they were different there?
Sure. We don't have access to that information. Our
Mhm.
donors are just from a general blood bank. So the only information that we get is their um, an STI panel and positivity or negativity and all that. That's the only thing we know about these individuals. It's a question that I
Okay.
do get asked a lot, actually.
Yeah, no, I mean, it is, because we know that there are differences, right? I mean, and would
Yeah.
expect that. You wonder if you could, if you were go, I mean, you could potentially, identify it by, just doing the PCRs for, for Y chromosome, for example, right? To see there's,
Oh,
um, to
I wouldn't
see,
have thought of that.
yeah. So just think about it. I mean, it might be, it might be worth doing it. And I was wondering if you have tried also any other HIV strains. I mean, I saw you did everything with ADA and what the MOIs, because the MOIs may make a difference too, right? In the
Yeah, sure. So we use ADA because it is a CNS, clinical isolate, and we're trying to, focus on, and it's an R5 strain, and we're
Mhm.
using macrophages, of course. Um, as far as the MOI, we always use 20 nanograms per mil infection, and that goes
Okay,
across the
just
board.
so,
I
but
will point
you
out,
standardize by P24 amount basically, right?
yeah,
Because
exactly.
yeah, okay.
I'll point out that we don't in the P24 that I showed all the way at the beginning, you see really, really big variability in donors. Some really just aren't very infected, and some are hugely infected. Um, but we always use the same MOI by P24 concentration.
Mm. Yeah.
Yeah.
No, it's really nice. I was just wondering, maybe some comment, because when we looked at mass and macrophages in the past, sometimes it takes longer, because you go for 7 days, we saw sometimes difference only occurring after 9 or 12 days, that the mass made a difference in
Oh,
terms
that's
of the
interesting.
production. And I mean, you could, if you, and also it's very nice what you showed to some of the stainings, you could, what we did in the past is, we were just staining the cells for P24. I mean, it's not 100%, but it's, you get some idea which cells are,
Yeah.
sufficiently, infected and actually producing, protein. And for the CD28, it was curious. Were those data that you showed with the up regulation, down regulation, those the same donors, so at different time points, or was it?
Yeah, so both the where I showed the maximal, the increase and decreases, that's the
Yeah.
same donors across different time points. Whereas for our other genes, we really didn't see a strong down regulation or a decreased expression in any other gene. But I noticed that and I said, hey, actually the sequencing data showed that for the 5 donors, it was a negative log twofold change value. I think both have biological relevance, but we're just still not really sure what to do with
But
the
it's
sort of
interesting because
sequencing there.
that seems pretty consistent. It would be interesting to follow up how that changes actually their ability to activate, right? Or in terms of also antigen presentation or T cell activity. Great, thanks.
Thank you so
Very, very
much.
nice work.
Thank you.
Fata, thank you. Fata, you had a question?
Yes, excellent, excellent job, Jessica. I especially loved your very last figure. I thought that was quite significant. I imagine that's a syncytia, is that right?
Yes.
Yeah, I love that. I think that was really nice. My question is about and again, this is not something that Joan is going to keep you for another 2 years as a postdoc for you
Well,
to
maybe
answer,
throw it out.
but are you thinking about the DNA damage response? Because these MT family members, they actually regulate a lot of the semi-naked DNA in mitochondria and also the nuclear DNA. And one of the reasons I ask is because I'm not convinced many times when we do infection in macrophages or even from patient material, whether the stuff is integrated or not, and whether we have a concept of copy numbers. We know the strain. I think that's very true, the type of virus. But at the end of the day, where does this virus live and coexist? Is it integrated? Is it not integrated? That's a separate question. But the overall DNA damage response in these cells, I'm just wondering if you're thinking along those lines.
Yeah, it's definitely a great point. I'll say that the LDH data that I showed all the way up top, I think suggests that meth isn't necessarily causing any cell death. But as far as DNA damage, it's definitely not something we've looked at, and it's really something to consider for sure.
Excellent. Thank you.
Thank you.
Jules? Okay. So why don't we go on right now, Cecilia?
Yeah, hi.
Oh, Jules is there. Sorry, I didn't see you.
Yeah, so I just had a couple of points. So it seems to me, and if you could confirm this, that the effect on inflammation probably increases the risk for all comorbidities due to meth, including heart disease and kidney disease. And also, you mentioned that meth might increase the risk for Parkinson's and Alzheimer's. So if that's the case, then it might confound the increased risk that could be found for Alzheimer's in older people with HIV. What are your thoughts?
I want to make, I think, a little bit of a clarification there. What I was showing is that in our in vitro studies of macrophages, when exposed to meth, the sort of groups of genes that were changed were those related to Alzheimer's and Parkinson's. We're not necessarily saying that meth use is exacerbating people's potential for developing those disease processes, but that the phenotype in the cells are similar to cells in those conditions.
I see.
Hope that helps.
Right, but still, that suggests that there might be an increased risk. Right? Or right?
I don't know necessarily. I think it just means that the cells are sort of behaving similarly to produce that kind of effect. I don't know, Joan, you have any thoughts there? But
I don't know that we can say that there are a lot of studies going on at the intersection of Alzheimer's and HIV, and so I think that more will come out as more people are being identified with Alzheimer's. So
Okay, then. Right. All right, but I think it's just something to think about then.
yeah,
And,
definitely. And it's always a great point. Thank you.
and then the other point is that the increase in inflammation probably affects all the other comorbidities as well, heart disease and so forth.
Absolutely. Definitely a really great point to emphasize too.
Thank you.
Thank you.
Jules. Cecilia?
Thank you. Thank you, Jessica. This is gorgeous. Appreciate your caution in many things. So thank you. And I'm a fan of reactive oxygen species. I love them. And I was a bit surprised that you didn't see much of a difference. And I'm assuming you are looking at 24 hours.
Yeah. So each day is a 24-hour time point.
Yeah. So I would be cautious there too, because reactive oxygen species, usually they start really as soon as you put meth.
Yeah.
And then they plateau within 4 hours. And then what you see is that the cell starts uptaking, and so it goes back down because ROS becomes a signaling molecule,
Yeah.
superoxide, and largely because it is related to what Fatah is saying, that it's a huge component in, modifying chromatin, which can contribute to, increasing transcription of some sets of genes. And so it's very cool. I think it's beautiful. Just, take a look at what happens if you block reactive oxygen species in this model, or just take a look a little bit earlier. You know, there are some dyes, I think is dihydroetidium, something like this, DHE. It's a live superoxide detector, and you can put there and you can see the cells living and you know, lighting up superoxide, or just kinetically take a little bit of supernatant. So you don't need to terminate the experiment to do this. So you can use, you know, it doesn't matter how many cells you get in your pack. You don't need to terminate the experiment. You just sample media to make a kinetics over there. It's
Cool.
gorgeous.
That's a great suggestion. Thank you so much.
You're welcome.
Yeah, I do just want to highlight, as you said, you know, ROS is definitely a very early sort of quick response thing. We were trying to, you know, sort of model the sort of, you know, is it daily meth exposure on like a regular basis? But you're absolutely right. That could be why we didn't really capture such a big response, is because it was probably earlier and then it's quelled. But
Yeah,
it is
but
interesting
it
to
doesn't kill the cell.
sometimes see the increase at
Yeah,
those later time points.
we always think that ROS is going to kill the cells. And in these cases, like with 50 micromolar meth, it's not going to kill the cells. They start reuptaking to use it as a signaling component. And this is a fascinating mechanism to me, and I love it. I think you have an opportunity to dig there.
Yeah, thank you so much for the comments and suggestions.
So there are more questions in the chat, but I think I'll leave you to look at them later because we're way over time already, and I know Leah has some last comments. So thank you, everybody. It's extremely helpful. Thank you, Jessica, for a beautiful talk, and thank you, Caroline, for, you know, all your hard work and organizing this year.
So Jessica, amazing job. And I just wanted to kind of come back and end this, because it was an amazing, and it was the last seminar of this kind of academic calendar year. And so please pay attention to an email from Caroline, likely either today or tomorrow, and it will have a link to the fall 2025 schedule. So people can see open dates and sign up to speak, as well as a link for the 2024-2025 recording, so people can view the seminar recordings from this past year. So please, if you want to speak, if your students want to speak, sign up, because this is when we, you know, we're trying to push so we have an amazing calendar set for next year. So thank you all. Happy Wednesday.
Okay,
Thank you all so much. This was such a great
Thanks, everyone.
experience. Thank you.
Thanks, Jessica.